Fouling community. A dock presents a great opportunity to investigate fouling communities. Animals in this community attach to the sides of piers, docks, and the bottom of boats. Here's how to collect fouling community organisms. Find an out-of-the-way spot on the dock to set up your workstation where you will not block foot traffic. Look along the dock for submerged surfaces that are heavily encrusted with marine life. On undisturbed surfaces, open space for attachment is usually hard to find, and many fouling organisms grow over one another. Typically, a fouling community consists of filter-feeding sessile animals. Do you recognize any of these animals? Hold a small bucket underwater and use a putty knife or spatula to pry off a chunk of encrusting organisms into the bucket. Do not collect where there are hydroids, small organisms that look feathery or plant-like. These sting when touched. If in doubt, check with your instructor. Slide the tool along the substrate, lifting off encrusted organisms. Do not hammer or scrape. If you want to view or take photographs of the organisms, you may place them in a small aquarium. Transfer the sample into a larger bucket. Because fouling organisms tend to accumulate a lot of muddy sediment, the sample should be rinsed with clean seawater. Pour the rinse water through a sieve so you don't lose any specimens. If possible, collect a second sample from an area with less light exposure. Try the shady side of the dock or reach farther underneath to remove a sample. Keep the samples submerged at all times. A cold pack keeps the sample cool while it is transported back to the lab. Next, you'll need to sort your specimens. Your group will work together to carefully pick apart clumps of fouling organisms and sort the different species you find into phyla. Mussels are often overgrown with slimy, rubbery material. These are compound tunicates, filter-feeding members of the phylum Chordata. Peel off a section and place it into a container of seawater. Clumps of mussels form a shelter for smaller creatures. To find them, pull apart the mussels one by one. Mussels attach to the substrate and to each other by protein strands produced by the byssal gland inside the shell. Sponges should be torn open and examined. You may find small crustaceans living inside the tunnels and cavities. Most solitary tunicates feel leathery. When you squeeze them gently, you see why they're commonly called sea squirts. Watch how this tunicate takes in the red powder. What has happened? Use forceps and plastic spoons to gently pick up small mobile organisms. Carefully separate mobile and sessile organisms from the sample and place them into containers of clean seawater. When sorting is done, lay out paper and group the specimens by the phylum to which they belong. You will organize and count the total number of each type of organism and compile your data. Then you can make comparisons about species diversity and abundance between the locations you sampled from. Write your groupings on the paper as shown in this example.